back to this video lesson on sex determination in organisms. The learning goals to be achieved from this session are: 1. Describe how sex chromosomes were discovered. 2. Elaborate on the chromosomal sex determination systems in organisms. 3. Differentiate between male heterogamety and female heterogamety. 4. Explain why a male honeybee has no father and produces no sons. And 5. Justify how climate change puts existential threats on sea turtles. Well, sex determination refers to a system to establish an individual or an organism to be a male, a female, or a hermaphrodite. Scientists have worked for hundreds of years in order to understand the sex determination systems in organisms. For instance, in the early 335 BC, Aristotle proposed that sex is determined by the heat of male partner during intercourse. If the male's heat is stronger than female's coldness, then a male child would be born. However, the first breakthrough in the understanding of sex determination came from the discovery of sex chromosomes during the early 20th century. In the year 1891, a German biologist named Hermann Henking, while studying spermatogenesis in firebug beetles, observed that although most of the chromosomes were present in equal numbers in both males and females, there was one darkly stained unpaired element in males. And he called these unpaired dark stained element as X element. X for an unknown factor, because he was unsure whether it was a chromosome. And he observed that male firebugs produced two types of sperms. One type of sperm with X element and another type of sperm without the X element. Later, in the year 1902, an American zoologist named McClung, while studying spermatogenesis in different insects, found that male insects had an unpaired chromosome. And this unpaired chromosome segregated unequally during spermatogenesis. So some of the sperms received the unpaired chromosome, while other sperms lacked the chromosome. And he termed these unpaired chromosome as accessory chromosome. He also hypothesized that the unpaired accessory chromosome are responsible for determining male sex in insects. Now, in the year 1905, an American geneticist named Nettie Stevens was working on mealworm beetles. And what she discovered was a pair of unmatched chromosomes. So it was an unmatched chromosome because one of the chromosomes was a small chromosome and the other chromosome of the pair was a larger chromosome. And around the same year, Edmund Wilson also discovered the similar small-large chromosome pair in the males of different insect species. And independently, this led to the discovery of the X and Y chromosome. So the larger chromosome was called as X chromosome and the smaller chromosome was called as Y chromosome. And they proposed that, Stevens and Wilson proposed that what McClung observed as accessory chromosome was the larger X chromosome, as well as what Henking observed as the X element was also the X chromosome. And these two X and Y chromosomes were collectively referred to as sex chromosomes because X and Y chromosomes are responsible for establishing with the sex of an organism. And hence the chromosomes are referred to as sex chromosomes. The above mentioned evidences and many more analyses of additional species over the years has revealed that sex chromosomes or chromosomes in general are responsible for sex determination in most of the animals. Let me now explain some of the chromosomal bases of sex determination in animals. The first one is XXX0 system of sex determination. In XXX0 system, XX organisms are females and X0 organisms are males. So in this system, females are having two two sex chromosomes, which are both X chromosomes, whereas males have only one sex chromosome, and which is X chromosome. They lack the second sex chromosome, which is denoted by 0. So 0 denotes to the absence or the lack of the second sex chromosome. Now, this type of sex determination system is very typical in cases of grasshoppers. So, the diploid chromosome number in grasshopper is 24 or 12 pairs. So, suppose we say that a female grasshopper with XX type of sex cells are going to be producing X cell. So, these are always going to be making X which contain only X chromosome. So, they will be producing only the same type of egg throughout their life. Whereas, male grasshoppers with X0 type of sex chromosomes are going to be producing two different types of sperms. Sperms containing X chromosome and another type of sperm containing no sex chromosome or zero sex chromosome. And because they produce two different types of sperms, such individuals or such sex is referred to as heterogametic. So in the XX-X0 system, males are heterogametic because they produce two different types of sperms, whereas females are homogametic because they only produce one type of egg. And by using the Punnett square, we can now determine how the combinations are possible. So the sex of a grasshopper depends on which sperm fertilizes the egg. So if an X chromosome containing egg is fertilized by an X chromosome containing sperm, then this would be resulting in X combination in the progeny, which is going to be a female grasshopper. And a female grasshopper would be receiving 11 autosomes and one X chromosome from the female gamete or the egg, and 11 of the other copy of autosome and one more X chromosome from the male gamete or the sperm. And this would be resulting in a female having 2n is equal to 24 chromosomes, or it is going to be forming here 11 pairs of autosomes plus 2 X chromosomes. So 11 pairs plus 2 is going to be 24 or 12 pairs of chromosomes. Whereas if an egg is fertilized by a sperm with no sex chromosome or zero sex chromosome, then this is going to be resulting in XO or X0 individuals. And X0 flies are going to be grasshoppers are going to be male grasshopper. And here they have one less of a chromosome because the second sex chromosome is lacking or absent in male grasshopper. So their 2n number is going to be 23. So they have 11 pairs of autosomes but only one X chromosome or one sex chromosome. And this system is known as XX-X0 system, typically in grasshoppers. Second system of sex determination is XX-XY system of sex determination, where XX individuals are females and XY individuals are males. Now in XX-XY system, both of the females and males have two sex chromosomes each. Okay, in females we have two of the X chromosomes, whereas in males one of the sex chromosomes is X and the other sex chromosome is Y chromosome. Now, XX-XY system of sex determination is a typical system that we have, as well as it is seen in many of the other mammals. So, females with XX type of sex chromosomes are always going to be producing one type of egg. All the eggs are going to be carrying X chromosome in it. Whereas, males with XY type of sex chromosomes are going to be producing two different types of sperms. One type of sperms have X chromosome in it, whereas the other type of sperms have Y chromosome. And because they produce two different types of sperms, males in XXXY system is again known as heterogametic sex. So males become heterogametic and females, because we produce only one type of egg, females are homogametic. Now in humans, as we know, our diploid chromosome number is 46 or 23 pairs. So we have 22 pairs of autosomes and 1 pair of sex chromosome, which is XX in case of females and XY in case of males. Now the sex of the baby, or the sex determination in humans, we need to keep this in mind, is chiefly dependent on the father or the male parent. Okay, that is because a gene has been discovered on Y chromosome, a gene which is named as SRY gene. So this is the name of the gene on Y chromosome. So SRY stands for sex determining region of the Y chromosome. And this gene on the Y chromosome would always be stimulating the development of testes in the embryonic stage. And that would be leading to the baby becoming into a male child. So whether an individual or whether a baby becomes a male or a female baby is dependent on which sperm fertilizes the egg. Okay, so now let's look into the Punnett square here. So we have two different types of sperms from the father or the male parent. So one sperm having X chromosome, the other sperm having Y chromosome. But we also know that since both of these carry the sex chromosome, so the kind of chromosomal combination that they are going to be bringing in is each of these sperms are going to be having 22 autosomes and one sex chromosome, which can be X or which can be Y. And the X cell are going to be having 22 autosomes and the X chromosome. Now, sex would be determined by which sperm fertilizes the egg. So if an X chromosome containing sperm fuses with the egg, then this would be resulting in XX combination. And we have to understand here that SRY gene is absent or not present on X chromosome. Okay, there is no SRY gene on X chromosome. And in the absence of SRY gene or in the absence of Y chromosome, by default, human sex development is always going to be a female. So this would be becoming into a female. Okay, whereas if a Y chromosome containing sperm fertilizes an egg, then this would be resulting in XY combination. And because we have the Y chromosome in the embryo, so Y chromosome, since it carries the SRY gene, is going to be activating the development of testes in the embryo. And thereby the sex of the baby would be male. Now if you look into the chromosome number, so 22 and 1 X from the egg and 22 and another X from the sperm is going to be resulting in this is going to be 44 of autosomes or 22 pairs of autosomes, and it's going to be having X2 X's. Whereas since we have the combination of 22 autosomes from each of the sperm and eggs and 1 X and 1 Y sex chromosome, so this would be having 44 autosomes or 22 pairs of autosomes and an X and Y sex chromosome, which is going to be again 46. So our diploid number is always 46. And that is how we need to realize that sex determination in a human is purely dependent on the Y chromosome. So Y chromosome or SRY gene is the sex determinant. Third sex determination system is ZZZW system of sex determination, where ZZ organisms are males and ZW organisms are females. Now in ZZZW system, both of the males and females have 2 sex chromosomes each. However, what we need to be noting here is a striking difference of ZZZW system to that of XXX0 system as well as XXXY system. In ZZZW system, males have two similar sex chromosomes. So, males have two Z chromosomes as their sex chromosomes, whereas females have two dissimilar sex chromosomes. One of the sex chromosomes is Z chromosome and the other sex chromosome is W chromosome. Now, because males have two Z similar sex chromosomes, they are always going to be producing one type of sperm. All of the sperms are going to be containing Z chromosome. Whereas, since females are having two dissimilar sex chromosomes, they are going to be producing two different types of eggs. One egg containing Z chromosome and the other type of egg containing W sex chromosome. And because females are producing two different types of eggs here in this ZZZW system, females become heterogametic, whereas males are homogametic. Okay, and such kind of ZZZW system is typically seen in cases of most of the birds. So if we take the example of chickens, in chickens we have the chromosome number, which is their diploid chromosome number is 78, or they have 39 pairs of chromosomes, out of which 38 pairs are autosomal pairs and one pair is the sex chromosome pair. So the sex of the birds is determined by which egg fertilizes the sperm. So the female decides the sex of the baby bird. Now, if a Z chromosome containing egg fertilizes a sperm, which is also carrying Z chromosome, then the sex chromosome combination is going to be ZZ, which would be a male bird. And since both of these gametes are going to be bringing in one set of the autosomes plus one of the sex chromosomes, so this is going to be resulting in 38 plus 38 autosomes, which is 76 autosomes, and one Z from the sperm and another Z from the egg, which is ZZ. And their 2n number is going to be 78. Likewise, if the W chromosome containing egg fertilizes the sperm, then the sex chromosome combination is going to be becoming ZW. And ZW sex chromosome combination is of the female sex. And they are going to be again having 38 plus 38, 76 autosomes, 38 pairs of autosomes, and two of the sex chromosomes which are dissimilar. One Z, which is from the sperm, and another W, which is from the egg. And their diploid number is also 78. So in this way, we have a system which is known as ZZZW system, which is in contrast to the XXX0 system as well as XXXY system. Fourth type of sex determination system is ZZ-Z0 system of sex determination, where ZZ organisms are males and Z0 organisms are females. In ZZ-Z0 system, males have two sex chromosomes which are both similar sex chromosomes. So both of the sex chromosomes are Z chromosomes, whereas females have only one sex chromosome. 0 denotes just like in X0 system. So 0 denotes lack of the second sex chromosome. So they only have one sex chromosome, and that sex chromosome is the Z sex chromosome. They don't have the second sex chromosome. Now, since males have similar types of sex chromosomes, they are going to be always producing one type of sperm, and the sperms are all going to be carrying Z chromosome. Whereas, since females are Z0 type of sex chromosome, so they are going to be producing two different types of eggs. One type of egg are going to be carrying Z chromosome, that is the sex chromosome, whereas the other type of eggs are not going to be carrying any sex chromosome, or they are having 0 sex chromosome. And because females again are producing two different types of eggs here, unlike X0 system, in Z0 system, females are heterogametic and males are homogametic. Okay, and this kind of Z0 system is typically seen in cases of many of moths. Now, One example of such a moth species is a moth having 60 chromosome in them. So their diploid chromosome number is 60, which means they have 30 pairs of chromosomes, out of which 29 pairs are autosomal pair and 1 pair is their sex chromosome pair. Now, again in moths, the sex of the baby is going to be determined by the female moth, or which egg fertilizes the sperm. So if an egg containing Z chromosome fertilizes a Z chromosome containing sperm, then the sex chromosome combination is going to be ZZ, and ZZ is a male moth. So each of the gamete from the parents are going to be contributing one set of autosomes. So they are going to be bringing 29 of autosomes and the sex chromosome. So because we have 29 plus 29, it's going to be resulting in 58 autosomes in total, or 29 pairs of autosomes plus a pair of sex chromosome, which is in this case, because it was fertilized by a Z egg, so it's going to be ZZ sex chromosome pair. And this is going to be resulting in a diploid number of 60 chromosome in the moth. Whereas if an egg containing no sex chromosome, or if an egg containing 0 sex chromosome fuses with the sperm containing Z chromosome, then that would be resulting in Z0 combination of sex chromosome. And Z0, as we know, are going to be female moths. And in female moths, they are going to be receiving 29 pairs of autosomes, 29 set of autosomes each from the male and female gamete, plus only one sex chromosome from the male gamete, which is the Z chromosome. Female gamete doesn't contribute or doesn't have the sex chromosome. So, because of which their diploid number, in case of female moth, their diploid number is 59. So, it is one less because they have one less of the sex chromosome. So, this system of sex determination is known as, in cases of many of the moths, is known as Z0 system. A fifth type of sex determination system is Haplodiploidy system, where haploids are males and diploids are females. Now the term haploid and diploid refers to the number or the set of chromosome in an individual. So haploid organisms are those who have only one set or one copy of each chromosome, whereas diploid individuals or organisms are those who have two sets or two copies of each chromosome. Now, most of the higher animals or organisms are diploid organisms, including humans. We have 23 pairs of chromosomes, which means to say that each of our 23 chromosomes exist in a pair. Haplodiploidy system of sex determination is commonly seen in social animals like honeybees, ants, and wasps. And in this example, I'm going to be taking Haplodiploidy system in honeybees. So in each honeybee colony, we can be expecting a single queen bee or a single fertile female. And the major function of the queen bee is just to reproduce. Whereas we can be also expecting hundreds of sterile females in the colony who are known as worker bees. And the sterile female worker bees, their work is to gather food and to do all of the housekeeping works. Whereas there are also several of the male bees who guard the colony, and these male bees are known as drones. So the diploid chromosome number in honeybee is 32. That means they have 16 pairs of chromosomes. So their haploid chromosome number is 16 and their diploid 2n number is 32. 2n denotes diploid condition, n or 1n denotes haploid condition or haploidy. Now, the sex of the offspring in honeybees is determined by the queen bee. So the queen bee, while mating with the males, can be storing all of the sperms in her body in a kind of a pouch-like bag known as spermatheca. And the queen bee, depending on the need of the colony, would be deciding whether she would be laying the egg in the fertilized form or whether she would be not fertilizing the egg. So queen bee can be laying two different types of eggs. Eggs which are unfertilized or eggs which are fertilized. So if the queen bee lays a fertilized egg, then each of such fertilized egg would be having two sets of chromosomes, one each from the egg and sperm cell. And because of which the diploid offsprings are going to be becoming females. Okay, and they are going to be becoming, in most of the cases, they are going to be becoming the sterile worker bees, who are going to be having the diploid chromosome number, which is 32 chromosomes. Whereas, if the queen bee decides to release unfertilized egg, so if she doesn't fertilize the egg in her body and just releases the unfertilized egg as such, then the unfertilized egg would be having only one set of chromosome. That is the only set of chromosome which is carried in the egg cell. And these kind of haploid offsprings are going to be spontaneously developing into the male bees. And the male bees are thereby haploid offsprings or haploid organisms having only haploid chromosome number. So their total chromosome number is 16 because they are haploids. Whereas the female chromosome number is 32 because these are diploid individuals. So in this way, we understand here that in the Haplodiploidy system, haploids are going to be becoming the males and diploids are going to be becoming the female. Well, to add on to the information, the process by which an unfertilized egg spontaneously develops into an embryo or into an organism, now in this case into a male honeybee, is referred to as Parthenogenesis. So we learned that Parthenogenesis is a special mode of reproduction, a kind of asexual reproduction where we have an unfertilized egg spontaneously, directly becoming into an embryo. Another very interesting fact regarding male bees is that male bees have no father and produce no sons, but they can have grandfather and grandsons. Now we need to have a little bit, this is a little tricky, but it is easily understandable. So male bees have no father because we know male bees are formed from an unfertilized female gamete. So male bees have no father and produce no sons. They produce no sons because if the male bee is to be fertilizing an egg, his offsprings are all going to be females or daughters. So male bees can only be producing daughters on fertilizing an egg. So he can never be producing any sons. But they can have grandfather and grandsons. Now how he can be having a grandfather? He can have a grandfather obviously because his mother, who is the queen bee here, is produced through fertilization. So to produce the queen bee, she has been contributed by a sperm from a male bee. And that male bee who contributed the sperm for the formation of the queen bee becomes his grandfather. And he can also be having grandsons. Now how he can be having grandsons? He can have grandsons because if this male bee mates with a queen bee, so if he mates with a queen bee and out of his daughters that he is going to be producing through fertilization, if some of the daughters are going to be selected as a new queen of another colony or a new colony, then she would be producing sons. She would be producing sons parthenogenetically, and those sons are going to be his grandsons. So in this way, we say that male bees have no father and produce no sons, but they can have grandfather and grandsons. Let me now explain briefly into yet another chromosomal sex determination, which is in Drosophila. And this system is referred to as Genic balance system. So in Drosophila, the sex is determined by the ratio of the number of X chromosome to that of the number of sets of autosomes, which is represented by X over A, where X denotes the number of X chromosomes and A denotes the number of sets of autosomes. So we learned that Drosophila has 4 pairs of chromosomes, 3 pairs of autosomes and 1 pair of sex chromosome. And we also realized that in our previous work by Morgan that males usually have XY chromosomes and females have XX chromosome. But what we need to keep in mind is that the system of sex determination is not similar to XXXY system in Drosophila. So in Drosophila, if X over A ratio is equal to 1. So if the number of X chromosomes to that of the number of sets of autosomes is equal, then it's going to be becoming 1, and such ratio would be giving rise to female flies. So female flies can have XX type of sex chromosomes, can also be having XXY type of sex chromosomes, or can also be having XXYY type of sex chromosomes. Okay, so I don't want to be going into the details of these things, but you need to realize that here Y isn't the male determining factor. Okay, so Y doesn't decides with the sex of the flies. So it is the ratio of X over A that determines the sex. Now, if X over A ratio is equal to 0.5, then such flies are going to be all males. So males can be typically having XY type of sex chromosomes, or they can also be having X0 type of sex chromosomes. Now, if the X by A ratio is somewhere between 0.5 to 1, then these flies are going to be called as intersex flies, or these flies are technically even known as gynandromorphs. Gynandromorphs because they can be having both of the sexes in the individual. Okay, and then we can also be expecting X by A ratio
lesser than
0.5. Then it's going to be becoming super male or meta male, and if the X by A ratio becomes greater than 1, then it becomes super females or meta female. So in this way, we have discussed with all the major types of chromosomal sex determination systems in animals. Apart from the chromosomal sex determination system, many other sex determination systems exist. In some of the organisms, genetics or chromosome doesn't have any role to play in sex determination. In fact, the external environment is purely responsible for the sex of such organisms. And this kind of sex determination is referred to as environmental sex determination system. For example, in some of the species, sex is determined by the temperature at which the egg is incubated during temperature-sensitive period. So the incubation temperature of eggs during this very highly sensitive, temperature-sensitive period, which is a short period in the early developmental stage, would be deciding with the sex of the organism. For example, in sea turtles, okay, in reptiles, eggs which are from lower temperature nests. So if the temperature of the nest during the temperature sensitive period becomes low, then at that time all these eggs are going to be becoming into male sea turtles. Whereas if the eggs are incubated at a higher temperature during the temperature sensitive period of these particular organism, then these eggs are going to be becoming into female sea turtles. So in this way we realize that environmental temperature or the nest temperature decides with the sex of sea turtles. Now, in this context, I would like to be also giving a connection of how climate change or global warming is causing a threat to these sea turtles' survival. So, climate change is causing rise in sea level, or it is also resulting in stronger storms, which causes the destruction of the beach habitats for these animals. And several of the studies have also shown that warming of the beach sand temperature because of global warming has resulted in an imbalance in the sex ratio in sea turtle population. So because the beaches sands or the nest of these sea turtles which are in the beaches sand have a higher temperature, so the eggs are all going to be becoming into female sea turtles, thereby resulting consequently causing a shortage of male sea turtles in the population. So this is how we realize that in some of the animals, purely it is the external factor or the environmental factor such as temperature which can be directly affecting with the sex of the individual. Likewise, we can be also having many other unique type of sex determination systems in other animals, and some of these sex determination systems are still unknown to us in many of the organisms. So with this, I come to the end of my video lesson on sex determination in organisms. I'll catch up with my next video lesson on genetic disorders.